Good afternoon. We shall be discussing our papers. Serum p-Glycoprotein and Monomeric C-Reactive Protein are elevated in Takayasu arteritis. Takayasu arteritis is a rare granulomatous large vessel vasculitis. The assessment of disease activity in patients with Takayasu arteritis is imperfect via conventional inflammatory markers such as ESR and CRP. We know that a population of circulating T helper 17.1 lymphocytes associates with active disease in patients with Takayasu arteritis. And these T helper 17.1 lymphocytes express p-Glycoprotein, which contributes towards the property of treatment refractoriness. Therefore, we explored whether serum p-Glycoprotein, which can be measured using ELISA as opposed to the cell populations which require flow cytometry, and monomeric CRP, which is derived from the pentameric CRP at the sites of inflammation, whether these can differentiate patients with Takayasu arteritis and healthy controls, and whether they are different in patients with active or inactive Takayasu arteritis. We also analyzed serial changes in these biomarkers in a subset of patients with Takayasu arteritis who are immunosuppressive naive before and after immunosuppressive treatment, and also assessed correlation of serum p-Glycoprotein with various circulating CD4 positive T helper lymphocyte populations, which are known to express p-Glycoprotein. So we identified that serum p-Glycoprotein, monomeric CRP, and CRP are elevated in patients with Takayasu arteritis when compared with healthy controls, and they moderately predict Takayasu arteritis versus controls on ROC curves. We also identified that CRP is elevated in patients with active Takayasu arteritis, whereas the ratio of monomeric CRP to CRP is elevated in patients with inactive Takayasu arteritis. Upon drawing the ROC curves, serum p-Glycoprotein moderately predicted inactive Takayasu arteritis, whereas C-reactive protein moderately predicted active Takayasu arteritis. Upon multivariable adjusted linear regression models to predict active Takayasu arteritis, it was confirmed that serum p-Glycoprotein is associated with inactive disease, and C-reactive protein is associated with active disease in Takayasu arteritis. And serial changes in these biomarkers following immunosuppressive therapy revealed that serum p-Glycoprotein and serum CRP were reduced following immunosuppressive therapy. However, when we excluded those patients who were treated with tacrolimus, which is known to be an inhibitor of p-Glycoprotein, the reduction in the levels of serum p-Glycoprotein before and after immunosuppressive therapy was no more significant. When we correlated the circulating T helper populations with serum p-Glycoprotein, we found a weak negative correlation of serum p-Glycoprotein with T helper 17.1 lymphocytes expressing p-Glycoprotein. We already know that this T helper 17.1 lymphocytes associate with active disease, and from this study, serum p-Glycoprotein is associated with inactive disease. So the key findings of the study were that serum p-Glycoprotein, monomeric CRP, and CRP are elevated in Takayasu than controls. Serum p-Glycoprotein and the ratio of monomeric CRP to CRP are associated with inactive Takayasu arteritis, whereas CRP is associated with active Takayasu arteritis. And serum p-Glycoprotein and CRP also decrease following immunosuppressive treatment, while the serum p-Glycoprotein levels negatively correlate with circulating PGP expressing T helper 17.1 lymphocytes. Thank you.